urine leukocytes. This is test to detect your leukocytes in urine. Sorry. The other name of this test is leukocyte esterase. This test, LE, detect any microbial infection in the body. The test depends on esterase method. The esterase is produced by the leukocytes. So to screen this test is used to screen gonorrhea and for amniotic fluid infection. The combination of LE test with the urinary nitrite test provide an excellent screen for establishing the presence of UTI. The positive result mean that there is above than 5 leukocytes by high HPF. HPF, it's a high power field because you see the leukocytes under the microscope, the high power field. If urine stand long time, leukocyte will lyse and more intense reaction occur. The false positive occur with vaginal contamination, presence of glucose, albumin, ascorbic acid, vitamin C. The false negative due to the large amount of oxalic acid, which can inhibit the reaction, the reaction of the esterase. The creatinine test, it is a degradation product of creatine and the creatine phosphate. It increase in the case of kidney disorder, thyrotoxicosis, and the sport practice. The indican test is a colorless organic compound. It increase in cholera when tryptophan metabolism increase. Tryptophan is first converted to indole, then to indican by bacteria in the gut. This indican, the end product, will then be excreted in the urine, and upon exposure to air, it is converted to an indigo blue dye due to oxidation by atmospheric oxygen. So the presence of this indigo blue dye can detect the presence of cholera. And as you can see from the figure, the chart of the results of this test. This slide for knowledge to see what is macroscopic screening. So you can see what is the type of the test, what is the range, and what is the disease associated with this test. And this slide is for knowledge. Identification of pathological, physical, and chemical urine constituents. Abnormal pathological constituents of urine either you detect by macroscopic or microscopic. The macroscopic is physical test and chemical tests, while the microscopic you investigate the pathological urine constituents for substances that normally don't exist in urine, like glucose, protein, proteins, ketones. How to detect abnormal constituents? Normally, a lot of labs use a urine strip. This strip is a filter paper on plastic which has a reagent coated on it on different pads. It gives a color when react with substance in urine. The produced color is compared with a chart color visually or mechanically assessed, as you can see from the strip. Results are reported as a concentration, as a small, moderate, or large, as a plus sign, as a positive, negative, or normal. You can see as well this method is rapid, easy, gives early indication and qualitative. Therefore, usually 3 or there are other confirmatory tests followed by this urine strip. Reaction in strip is affected by time. To reduce timing error and to limit variation in color interpretation, automated machines is used to read the reaction color on each test pad, as you can see from the figure. This slide for knowledge, you can see the different urine strip for different tests. You can see a test for a specific gravity. You can see a test for ketone, glucose, nitrite, leukocytes, and pH. Urine analysis to show some how urine strip is very helpful in showing the disease. If you can see, this is a urine strip, the normal one versus after the test, to see to investigate. In this case, if you compare both of them, you will get to the result that this is a very diluted urine, and request avoided urine in the morning if persisting low SG. Possible it have a diabetes insipidus, and a microscopic evaluation may give a negative result. In this case, the same. So you need to go all through these cases to understand. You don't need to study or memorize, but to see the effect, the variation in the results of the urine strip analysis results, to see how these relate with the type of disease. Again, you don't need to study this slide, but only to understand. For this, for instance, you can see this are the feature, and this are the microscopic examination, and relate to type of disease, which is bacterial invasion of renal parenchyma. Another test would show a nephrotic syndrome, the presence of oval fat bodies, fatty casts, and waxy casts. Okay, so if we continue the microscopic analysis of urinary sediments, the methodology: normally a sample of well-mixed urine will centrifuge in a test tube at a very low speed. The supernatant is decanted and 0.3 ml is left on top of the sediment. The sediment is resuspended in the remaining supernatant by flicking the bottom of the tube several times. And you took a drop of the resuspended sediment, is poured into glass slide and covered with lip. This slide is very important to dissect all the microscopic analysis of the urine. You take the sediment, you check for cells, cast, crystals, organisms. You relate this to a disease associated, either glomerulonephritis, nephrotic syndrome, pyelonephritis, lower UTI. You give an image index, and you have a final exam. So this is a very good slide to summarize all the microscopic analysis.